So we're starting the day 2 video of lesson 7.3. Yesterday we talked all about diffusion and how diffusion is a type of passive transport, which means that it requires no energy. The reason diffusion is a type of passive transport is basically because substances are moving from an area where they are in a high concentration to an area where they are in a low concentration. So a lot of substances can undergo diffusion. However, not all substances can pass straight through the membrane. So if I look at this, small uncharged molecules like water, carbon dioxide, oxygen, nitrogen, ethanol, all of these, they can pass straight through the membrane and go right in, no problem. But when it comes to other substances like sodium, chloride, amino acids, and glucose, all vital substances which the cell needs, they can't just go straight through the membrane through diffusion. So instead, they need to undergo facilitated diffusion. The word facilitated means helped or aided. So for example, if I say that I'm going to facilitate a process, that means that I'm going to help a process to happen, right? So this can still be considered diffusion, but the reason it's called facilitated diffusion is because it requires no energy. You are still going from an area where you're in a high concentration to an area where you're in a low concentration, which means that this is a process that naturally wants to happen. The only difference is that it's facilitated, meaning that there is going to be a protein involved. So now, when I'm looking at facilitated diffusion, I can see that there has to be a protein. This is just regular old diffusion. These little molecules are moving straight through. See these purple ones? These might be charged molecules, or they might just be too big to squeeze through, so they need to go through an actual protein, okay? Diffusion and facilitated diffusion are types of passive transport because they require no additional energy, because they move from areas of high concentration to areas where they are in low concentration. So you see here how there's 5 of them, here there's only 2, so naturally they would want to pass through here and enter this area. If I was trying to move this one from here, where it's at a low concentration, out here, that would require energy because that would have to be something that needs to be forced to happen. Okay? All right, so now let's talk about the different types of proteins. So we have channel proteins, and then we have carrier proteins. A channel protein is a protein that just is open. It can open and it can close, but it doesn't tend to change shape all that much. Once it's open, anything can pass through as it pleases. And when it closes, all movement stops. A carrier protein, however, is a protein that changes shape to allow substances to enter. So let's look at this carrier protein. It starts off like this, where it's open on the outside of the cell membrane. It grabs one of these, and then it closes in on it, and then it opens up on the other side. So it's not a full opening where substances can just move in as much as they want. They have to move in piece by piece. Enter, close, exit. Enter, close, exit. This is a carrier protein because it carries the substance into the cell by changing shape. A channel protein is basically just an opening. It's a door and things can pass through it. Now, even though when we're looking at channel proteins and carrier proteins, even though they look like they can let anything in, that's not actually true. They are very specific to which substances can enter. So if you see this carrier protein, it can only let in these little oval-shaped molecules, while the channel protein is only allowing these little hexagons to actually enter. So let's go ahead and try this experiment right here. All right, so you guys, this is a PhET experiment. It is open for you. However, to use this, you will need to download Java. So I'm going to just do it with you. And if you have Java, you can go ahead and do it on your own. So let's go ahead and fill up the cell with a whole bunch of these blue molecules. You can see here that these blue molecules are not able to permeate the membrane. So you see this? This is the membrane. They cannot diffuse straight through. They are trapped here on the outside of the cell, right? So let's go ahead and let's put in a channel protein and see what happens. See? Now they can enter the cell. And I can get more and more and more channel proteins depending on what the cell needs. So let's say this is the inside of the cell and it needs a lot of the little blue ones. This means it will create more channel proteins and it will allow a lot more of the substance to actually enter. And when you look over here, you can see that the concentration of blue here is high, the concentration of blue here is low, and they're going to keep passing through until they reach a point where they're equal on both sides. Okay? So now let's clear all particles and let's just reset this whole thing. Yes. And let's go back here. I want to now put a whole bunch of blue ones, right? And I'm going to put a gated channel here. Okay? Now what a gated channel does, I'm going to put the concentration up here, and we're going to look over here. See how here the concentration of the blue diamond substances are very high and the concentration of blue here is very low? When I open this gate, substances can enter. So now the cell has what it needs. And as soon as it has as much as it needs, the gates will close because it doesn't need any more. This is just another way that cells can regulate how much is coming in and how much is leaving, right? So if the cell needs more, it can open up again and more of them can enter, and they will close when the cell has had enough of it, right? All right, so now let's see what happens when we start to mix in a few green ones. I'm going to put in one of these, and I'm just going to open one of these. So I'm going to fill up, or you know what, we'll keep these closed. I'm going to fill this whole area with blue. So I now have an open channel. Let's see what happens. See, the blue ones cannot enter. This is because channels are extremely specific to what they allow to enter. So let's put in a few of these green ones and let's see where they go. Now, they are at a high concentration up here, so naturally they want to move in here. So you see this gate is wide open, but it only allows the green substances to enter. And this is exactly how it would work inside of your cell. Different channels are made for different substances. If I have a channel that looks like it's wide open, the green ones cannot go in through here because that is not possible. It is made specifically for one type of substance, while this one is made specifically for another type of substance. So let's say I need a lot of the green particles. My cell will make a lot more of these, and it will close up the blue ones to allow a lot of green in and to stop the blue from entering. This is just another way that your cell regulates what enters and what exits, okay? And as you can see, this is a natural process because these molecules are moving from areas where they are at a high concentration to areas where they're at a low concentration, right? All right, so we're gonna go back here. And that's the basic idea. This is called facilitated diffusion because it's still diffusion. Substances are going in a natural order. They're going from an area where there's a lot of them to an area where there is not a lot of them. Because remember, substances or materials or compounds, they are like antisocial people at a party. They just want to get away from the crowd. They're trying to go from an area where there's a lot of them all squished together to an area where they can have more space. This is why they naturally want to move this way, and this does not require energy because they want to go this way. And they can move into a cell either through diffusion, but of course, this is not the case that we're talking about today, or through facilitated diffusion. Facilitated diffusion can use either channel proteins, which are wide open and can close, or carrier proteins, which can allow substances to enter, close, and open on the other side. So they have to change shapes to allow substances to enter the cell. All right, so now guys, you can go ahead and try out that experiment on your own if you can get it to work. Otherwise, this video will be enough. And then you guys can go ahead and work on your quiz. Of course, if you are confused, you can come back and replay this video. And if you really are confused, you can message me on Shobi. All right, guys, good luck.